the fungi. So far we have studied about the different types like Rhizopus and a typical example, and basically we have studied the general characteristics of fungi, the thallus organization and the reproductive methods. So now today we shall study about the next example, that is Pencilium. So Pencilium, it is also referred as Talaromyces, another name for that. So Pencilium notatum and various species are there in regarding about Pencilium. Now this Pencilium, it is purely a saprophytic fungus. So but however there are certain species of Pencilium which are parasitic and they cause various diseases. So this Pencilium is commonly called as green mold or blue mold. So Rhizopus is called as bread mold. Pencilium is called as green mold or bread mold or blue mold. Now this is because of the appearance what they are seen. So as I said, it is purely a saprophyte, and it is present on decaying fruits, and just like Rhizopus and also vegetables. So we find Pencilium growing on vegetables and also on the decaying fruits. There are species of Pencilium which cause disease. For example, Pencilium italicum and Pencilium digitatum. These two species, they cause a disease called as soft rot in citrus. And there is another species of Pencilium like Pencilium funiculosum, which causes a fruitlet core rot disease in ananas, pineapple, and these are some parasites examples. Now there are certain species which help in fermentation of cheese. For example, Pencilium roqueforti, Pencilium camemberti are two species which cause fermentation in the cheese, and they help to produce a characteristic flavor or characteristic aroma to that particular cheese. So this is about the occurrence or the Pencilium distribution. Now if you come to the thallus, so thallus means, as we know that fungi are made up of hyphae. Similarly, Pencilium is made up of hyphae. So except yeast, so Pencilium belongs to ascomycetes, whereas Rhizopus belongs to phycomycetes. So ascomycetes, they are sac fungi, and these ascomycetes, which are called as sac fungi because of the ascus which appear like sac reproductive structures. So this Pencilium is also made up of hyphae, the hyphae which forms the Mycelium. So this naturally when we see the Mycelium, it is a colorless slender and tube-like structures, and which are branched and they are septate. So the Mycelium is septate. So suppose this is the hyphae, so this is branched. So and whereas in Rhizopus we find aseptate, there are no septa, the nuclei are freely distributed. That is why it is called a cenocytic condition. But in case of Pencilium, it is having a septate hyphae, and so the cells are having nuclei. The cells may be uninucleate, the cells may be binucleate, or the cells may be multinucleate. So this is because as we have studied in the general characteristics initially in the starting part of this unit, whereas though the hyphae are septate, but the septum is not complete. There is a small pore in the center. So in the center there is a small pore. Though the partition is there, there is communication through this pore, and the cytoplasm or sometimes even the nucleus from one cell may enter into another cell. So when it enters that cell which receives the nucleus, it appears to be binucleate. So that is why the cells may be uninucleate, binucleate, or sometimes they may be multinucleate, means they may be having more than single nucleus. So this is about the hyphae. Now the hyphae, whenever it is present in a substratum, for example, if you take in a fruit or if you take in the vegetables, so here the hyphae penetrates the substratum to absorb the nutrition. So that nutrition, the hyphae which absorbs, suppose here this is the hyphae which absorbs the nutrition, so this is called as hostorial hyphae. Hostoria, they are the structures for absorption, isn't it? So hostorial hyphae, they absorb the nutrition from the substratum, and they are the hostorial hyphae. Now there are certain hyphae which are aerial. They are the structures which help in reproduction. So like that, the hyphae may be aerial, or sometimes they may be in the deeply growing into the substratum. Now such hostorial hyphae, they secrete enzymes, and these enzymes they help in the absorption of the food materials from the substratum. So this the septal walls, the cross walls are having a central pore through which the cytoplasm is communicated from one cell to the other cell. That is how the mycelium is present. Now if you come to the structure of a single cell, so here though the cell may be nucleated, uni or binucleated, but here the cell wall, if you take, the cell wall is thin, but however it is made up of 4 layers here. So there are 4 layers in the cell wall. So suppose just to mention here, suppose these are the 3 layers. So here the 4 layers which are present. So what are there? Now the first layer, that is the outermost layer. Suppose if you take, this is the outermost layer of the one, it is made up of a material called as glucans. Suppose this is one, and this is the second one, third, and this is the fourth. So now the outermost layer is made up of a carbohydrate polysaccharide which is called as glucans. But the second layer is a protein layer. It is made up of a simple protein which is the part of the cell wall. And the third layer is made up of a structure called as fibrillar electron transport. So that is the third layer of the cell wall. And the fourth layer is made up of pectin or hemicellulose. Sometimes it is made up of pectin, or sometimes it is made up of hemicellulose. So these are the 4 layers which we come across in the penicillium. So the cell wall is made up of 4 layers. The outermost glucans, the second inner protein, the third fibrillar electron transport, and the fourth pectin. Now in the next part of the chapter, so here the cells, I said it may be uninucleate, or it may be binucleate, or sometimes there may be 3 nuclei also. It is because of the movement. So 3 nuclei is possible whenever a nucleus from this cell and this cell enters into a single cell. That is why there is a trinucleate condition, and in the cells oil globules are present as a reserve food material. That is about the vegetative structure or the structure of a single cell. Now the remaining parts, there are cell organelles, cytoplasm and all that. In general, characteristic feature is only about this cell wall and the presence of single nucleus, two nuclei or three nuclei, uninucleate, binucleate or trinucleate. Now if you come to the reproduction in Pencilium, so Pencilium reproduces both by asexually and sexually. So the asexual reproduction takes place by vegetative type of reproduction and sporulation. So this asexual and sexual main. So asexual takes place by vegetative method and sporulation. So vegetative method is only by fragmentation. So fragmentation is one method, and sometimes it is also taking place by the help of the sclerotia. Now in case of vegetative type of reproduction, here fragmentation, it is a common method as we have already studied in the previous case. So fragmentation is the separation of a segment of a hyphae in the mycelium, and this separation of the segment or a part of the hyphae, it does not degenerate or it is not lose its existence. So then it divides the cell which are present in that part of the mycelium or the segment. So each fragment, it divides and produces a full-fledged mycelium or a completely well-developed body of the Pencilium. Then in case of sometimes what happens in certain species, not in all, the mycelium, it forms a compact structure. So these hyphae, they closely form a compact structure, a body-like structure that is called as sclerotium. So these sclerotia, they are somewhat like rhizomorphs. These compact mass of mycelium, which are spherical bodies, so they act as resting bodies and they are called as sclerotia, and then they also help in the vegetative reproduction. Then if you come to the spore formation, sporulation, so this sporulation takes place by the formation of spores, which are called as conidia, produced in the hyphae. So this conidia, they are produced, they are non-motile spores which are produced in certain branches of hyphae. So the hyphae, certain hyphae, they give rise to structures, and as the cells are partitioned like this, so they may be branched at the tip, and this tip of cells, they produce the conidia. So these structures which produce the conidia, they are called as conidiophores. So a conidiophore is a hyphae which arises from any cell of the mycelium. So in a mycelium, any cell may give rise to this conidiophore. So conidiophores are vertical structures. So they are long, erect, and they are a special type of septate hyphae which are called as conidiophores, and these conidiophores they arise from any cell from the mycelium, and they are always vertical in nature. That means they are negatively geotropic, away from the substratum, and then they branch into primary, secondary or sometimes tertiary. So this branches like this, so into primary, or even it may be produced into secondary branches like this. So the branches, that means the conidiophore may branch into primary, secondary or tertiary branches. Now each branch at its terminal end, it produces a structure called as sterigmata. So here a structure called as sterigmata is produced. So they are somewhat flask shaped structures like this. So here these are all septate. So these are called as sterigmata, which are produced at the ultimate tips. Now below that the cell which is present, now this is called as medulla, or sometimes it is also called as phyalides. So phyalides, they are called, the cells are called as medulla. So this sterigmata are called as phyalides. So sterigmata are the terminal tips of the conidiophore, which are somewhat flask shaped structures. So these flask shaped structures, they are called as the sterigmata. So the sterigmata are uninucleate, and these sterigmata, they are uninucleate, and they produce the uninucleate conidia in the form of chains like this. So suppose if this is another sterigmata, if you take here, so these produce the conidia or the conidiospores in the chains. So in the B.Sc genus, means from the base, at the tip there are old spores, at the base there are young spores. So in a chain, hundreds of conidia are produced. So the conidia, they are separated. So these conidia, they separate. So these are the conidia. So they separate from the sterigmata, and they are carried by wind or by some other agents of environment like water and like that. So when the spores, they fall on a suitable medium. Suitable medium means the decaying fruit, vegetables or like that. So when they fall on a suitable medium, so they grow into a new mycelium. So the spores, they germinate into a tube-like structure, and the cells go on dividing. So the spore divides into two cells and form a tube-like structure, and which further divides and forms a branched mycelium on that suitable substratum. Then if you come to the sexual type of method, in the case of Pencilium. So sexual method means definitely there must be male and female. So here instead of this here also we come across the antheridia and the archegonia, which is called as ascogonium. So the antheridium is the male reproductive structure, and the female ascogonium, which is produced in the hyphae. So here the Pencilium species are homothallic in nature. So they are homothallic actually, and thus the hyphae, a particular hyphae produces antheridium and an ascogonium. So this antheridium, suppose this is the hyphae. So any cell of the hyphae produces a simple antheridium like this, which is having a single cell. So the antheridium, suppose this is the antheridium which is produced by the hyphae. So this antheridium, it is a short club shaped structure, and it is produced at the terminal part of the any hyphae. So any hyphae produces the antheridium at its tip, not in the middle. and this antheridium which is produced, so the male branch is somewhat inflated and it is separated as antheridium by a septum. So there is a septum there and this separates as the antheridium. That is how the antheridium is produced. Now if you come to the ascogonium, so the ascogonium, I said in the case of the Pencilium, usually it is homothallic, means in the same mycelium we find the production of the antheridia and as well as the ascogonium. Ascogonium means it is a female reproductive part. So this is female reproductive part is somewhat long than that of the antheridium. So this is the ascogonium. This is the ascogonium. So the ascogonium, it is also a septate. It is having a septate hypha, and this is very long, and it is having a multinucleate. So there are many nuclei in the ascogonium like this. And the ascogonium, it is tubular, it is not having septa, but it is having free nuclei, cenocytic in other words. And here the this ascogonium, it is having a somewhat umbrella-like structure. So the ascogonium at its upper end, it may be curved into a handle-like structure. Sometimes the tip of the ascogonium, it is bent like this, like a umbrella shaped structure, or may not be. We find such kind of structure in case of ascogonium. So then the ascogonium, which contains multinucleate. So how the reproduction takes place? So the antheridial branch comes in contact with the ascogonium. So the antheridial branch, it contacts the ascogonium, and here the wall of contact. So this antheridia grows towards the ascogonium and comes in contact with this ascogonium like this. So when it comes in contact, so here at the wall of the contact, so there is the dissolution of the wall. So at the wall of the contact, a pore develops where the two walls come in contact. That wall dissolves and a pore or communication develops. So the contents of the antheridium, that is the nucleus, male nucleus, enters into the ascogonium. So when the two protoplasts come in contact, and in addition to this, there are some sterile hyphae which also surrounds this antheridial and ascogonial branch. So this ascogonium, which becomes the sterile hyphae, it just surrounds the fused antheridium and ascogonium. So the ascogonium becomes septate, and each cell, so like this, the ascogonium becomes septate like this, and each cell, what happens, it contains a pair of male and female nuclei. So that means the male nuclei of the antheridium, it contains in one cell. Similarly, another antheridium enters like this, the fusion takes place. So there is a pair of nuclei in each cell. So one is the male nucleus and another is the female nucleus. So each cell in the ascogonium contains a pair of nuclei, one male nuclei and the female nuclei. Then in the ultimate and penultimate cells of the ascogenous fuse to form diploid nuclei. So in the tip of the cell and in this, they fuse to form a diploid nucleus. in the ultimate cell, that is in the tip of the ascogonium, and these cells and the penultimate cells of the ascogenial hyphae, they also fuse and they are called as the ascogenous hyphae. And these ascogenous hyphae and these are called as the ascus mother cells. They are called as the ascus mother cells. So each cell is called as the ascus mother cell. Now this each cell where the nucleus fuses, it divides 3 times, so to form 8 nuclei. So each ascus mother cell divides 3 times to form 8 nuclei, and out of this 8, 4 are of plus strain and 4 are of minus strain. That means male and female. So this antheridia are the plus and the ascogonia are the minus. So that is one which is used. And the hyphae which surrounds this ascogonial ascus mother cells, so it is called as peridium. So this peridium is the protection of this antheridium and as well as ascogonium. So this structure which is formed, it is called as the ascocarp, the complete spherical structure. So the sterile hyphae which forms the peridium, which covers the ascogonium, so that forms a spherical body called as ascocarp, and the ascocarp is closed by this sterile hyphae, and this is called as the clistothecium. So sometimes this is referred as the clistothecium. So this ascus which are formed inside, so these ascus, they are formed in chains within the ascocarp, and from this you will find the ascus like this from each ascus mother cell, and whenever the clistothecium breaks, so at complete maturity it will burst actually. Now after the clistothecium burst, the ascospores which are formed, they are liberated from the ascus and they are carried by the wind. So when these ascospores, they are fall on a suitable media, and also when there are favorable conditions, so sometimes what happens, even though the medium or the substratum is suitable, but the conditions are not favorable. For example, moisture and temperature is very important for the germination of the spores. When the temperature is not suitable, when it is a severe cold or sometimes very low temperature, and whenever there is not a sufficient moisture, when there is a dry air, so the spores will not germinate. So suitable moisture and temperature is necessary to germinate of the ascospores, and thus the ascospores will develop into a mycelium. So the plus type of ascospores and the minus type of ascospores, so they develop into the respective mycelium and develop into a new hyphae. So that is how the reproduction in case of Pencilium takes place. So Pencilium notatum is the common example which we come across, and so this Pencilium is septate, having hausturial hyphae and the erect hyphae which is called as the aerial hyphae, and then the cell wall is characteristic of 4 layers, and then asexual by vegetative and sporulation, vegetative by fragmentation and sclerosae formation, and sporulation by the formation of conidia. And whereas the sexual there is homothallic, and thus it produces the antheridae and ascogonia to produce the ascus mother cells, ascus and ascospores. So that is the reproduction in Pencilium.